Hello, everyone, and welcome to this Genetic Engineering and Biotechnology News webinar. It's entitled Accurate Prediction of On- and Off-Target Cardiotoxicity with Human iPSC-Derived Cardiomyocytes. One of the leading causes of failure for new drug compounds has been the lack of appropriate in vitro models that fail to recapitulate important aspects of cardiac biology. The ongoing development of induced pluripotent stem cell technology carries great promise for early predictive detection of cardiotoxicity. When combined with current high-throughput screening methods in pharmacogenomics, we may soon realize the full potential of personalized medicine. Those are among the topics our panel will address today, so let's meet our speakers. Blake Hansen, product manager at CDI, will introduce us to some of the basics behind the development of the iCell-cardiomyocytes and the different applications for which they have been utilized. Dale Baker, research scientist of exploratory toxicology at Celgene, will discuss data from studies on hERG cardiac ion channels, along with their usefulness in the drug discovery process. Matt Peters, principal scientist of discovery technology at AstraZeneca, will describe his work on developing sensitive cardiac toxicity screening methods using stem cell-derived cardiomyocytes. Before our speakers get started, I want to encourage you to submit questions for a Q&A session at the end of the presentations. We'll try and answer as many as we can. Simply type your question into the Q&A box on the left-hand side of your screen and then hit submit. So if everyone's ready, let's get the webinar going. Blake, it's all yours.
All right. Thank you, Jeff. And thank you, everybody, for joining us today. As Jeff mentioned, we'll be talking about using stem cell-derived cardiomyocytes in toxicity testing, specifically iCell-cardiomyocytes. I'm going to keep my part very brief, and the agenda for my talk will simply be looking at an overview of cellular dynamics, the company, iPSC technology, then moving into iCell-cardiomyocytes, as well as then really just a brief look at the advantages of using human stem cell-derived cardiomyocytes. You'll hear more about the specific examples and advantages from the following speakers. So to get started, Cellular Dynamics is the world's largest provider of stem cells and stem cell-derived tissue cells. Really, we've accomplished this by focusing on four competencies. That is the generation and culture of these cells, genetic engineering for suitable endpoints, developing new differentiation protocols to make new cell types, and then finally producing these cell types at an industrial scale. In order to do this, we've developed an extensive IP portfolio and have really assembled probably one of the world's finest staff at culturing, creating, and differentiating these cells. We refer to this as the stem cell operating system, and our products can fall into two separate primary buckets. One is our vanilla off-the-shelf iCell products, which we see here, a listing of a variety of cell types, as well as in a custom manufacturing production where we will take in donors or samples of choice, be it from diseases, clinical trials, or just special populations of interest, and then make the cell types of choice for you, the customer. All right. Really, the way we do this, to center on the technology that we're going to be talking about today, I want to run through this slide very briefly. This is induced pluripotent stem cell technology, where we have acquired a sample. That somatic cell sample, typically a blood sample, is reprogrammed into stem cells. Those cells are then expanded and banked into a large master and working banks. From each working bank, then, you can derive the various cell types of choice. Each new manufacturing batch will go back to that working cell bank, and so the real advantage here is that you're eliminating batch-to-batch or experiment-to-experiment variability while being able to introduce population or genetic diversity. And one of the advantages of cellular dynamics is that we have scaled this up, as mentioned before, so we can make literally billions of cell types or billions of cells from a specific cell type. This enables the end user to have high-quality, highly consistent product on demand. All right? We're going to really focus on the stem cell-derived cardiomyocytes today. So iCell-cardiomyocytes are a cryopreserved product that, when thawed, form a fully functional syncytial monolayer of cardiomyocytes that beat spontaneously. As you'll see, these cells exhibit the normal human biology, and they're really the industry standard. where they've got a broad platform utility, a very large literature database, and they're really being used by the majority of the top pharmaceutical companies. All right, this will be the only characterization slide that we present, and just suffice it to say that these cells, cardiomyocytes, have the appropriate genetic expression profile. Their protein expression profile is one that subserves their function. That is, you've got sarcomeric formation so they can contract. The metabolic activity mimics that of the adult cardiomyocytes, where it's mitochondrial-based but can become glycolytic under appropriate conditions. The biochemical pathways are all there, so one can look at GPCR and kinase pathways, and you've got the appropriate functionality. That is the translation from electrical events down to mechanical or contractile activity. Really, it provides a fully functional human cardiac prep that allows easy translation to the native condition. I think the real advantage of using stem cell technology, and specifically human stem cell technology, is that it provides a contextual relevance for your experiments. That is, in the case of structural toxicity, you're looking at that structural toxicity in a metabolically and mechanically active cardiomyocyte. Similarly, for functional toxicity, you've got the appropriate functional pathways, electrical to calcium signaling to mechanical activity. And one of the real advantages to using a prep like this is that you've got that contextual relevance, where functional or mechanical activity can exacerbate structural toxicities, and structural toxicities can affect functional activity. So you've really got the appropriate feedback and interplay. You'll hear a couple of great examples of how the cells are being used, but I also refer you to our citation index as well, where we can see well over 40 peer-reviewed publications that can cover both toxicity testing as well as drug development. In addition to really being customer qualified, these cells are also garnering the attention of the regulatory agencies, where they're being fully used in developing the new comprehensive in vitro proarrhythmia assay, as well as starting to be included in some of the IND filings that have been submitted by customers. So all in all, we're really seeing the iPSC technology come into its fruition, and we'll hear more about that shortly. So with that, I say thank you and turn it over to the next speakers.
Blake, thank you very much. That was a great presentation to get us started, and you provided us with some excellent insight on the versatility of iCell cardiomyocytes. Thank you. Before we move on to Dale Baker's presentation, let me again invite you to submit questions for our Q&A session at the end of all the presentations. Simply type your question into the Q&A box on the left-hand side of your screen and then hit submit. The speakers will try to get to as many as possible. Our next speaker is Dale Baker, and he is ready and raring to get going. Dale?
Thank you, Jeff. So the title of my talk is HERG eCypher Cardio, which is a multi-electrode array assay, and dog telemetry, and it's a retrospective analysis. So just a quick outline of my talk. I'm going to give an introduction, HERG background, an overview of the HERG assay as we know it, our compound X background, and yes, because of legal, it's compound X. Our eCypher cardio assay, which is our multi-electrode array assay, the dog telemetry study, a summary, and conclusions. So let's get started. So cardiovascular toxicity is a potential side effect of many discovery and development compounds. Additionally, cardiovascular toxicities have been a major reason for many post-market withdrawals. A lot of those fun players are Seldane, which is terfenadine, which we actually use as a positive control in a lot of our cardiotox assays, Bextra, which is valdecoxib, and Vioxx, which is, of course, everyone knows. And, of course, early evaluation of cardiovascular toxicities is a priority of both regulatory authorities as well as biopharmaceuticals and pharmaceuticals industry utilizing in vitro and in vivo preclinical models. So before we go on, I wanted to go over a little bit about the cardiac electrical activity and a primer. So in the heart, the atrial depolarization event is initiated by the sinoatrial node, which causes the P wave, and you can actually see that as the ECG wave on your right. Next, the atrial depolarization completes, and the impulse gets delayed at the AV node, atrioventricular node, and that's that pause between the P and the Q wave there. Once that happens, ventricular depolarization begins, and that's actually the QRS wave here, causing the QRX complex, and then atrial repolarization occurs. After that, ventricular depolarization completes, and actually this is the pause between the S and T wave. And then once this occurs, we actually have ventricular repolarization beginning at the apex, causing the T wave. And the T wave is actually what we're really interested in some of the next slides, and I'll go over that a little bit more. And then the next thing is just actually the pause after the T wave, actually after the repolarization completes. So back to my introduction slide, many of the cardiovascular toxicities are a result of blocking cardiovascular ion channels such as sodium, potassium, and calcium. And one such ion channel that we're really interested in is a potassium channel called hERG. So the hERG channel, for a little bit of background, is actually the human ether agogo-related gene, and it's a gene that codes for a protein known as Kv11.1, or a potassium ion channel. And hERG contributes to the electrical activity of the heart that coordinates the heart's beating by mediating ventricular repolarization of IKR, which is the inward rectifier, rapidly delayed rectifier current, in the cardiac action potential. Inhibition of this hERG channel can result in long QT syndrome or cardiac arrhythmias such as torsade de pointe, which literally means twisting of the points, which can degenerate into ventricular fibrillation. And below you'll see actually regular rhythm followed by TDP at the end here, where it degenerates into this ventricular fibrillation. So when we look at hERG blockade at a molecular level, cellular level, and tissue level, it's actually very interesting, the chain of events. You can actually see at molecular level, we have the drug inhibiting the hERG channel. We actually, at the cellular level, we actually have an inhibition of the action potential. We see here the black line is a normal. The red line is actually inhibition of the hERG channel. You see this inhibition of the hERG channel there. And then when you actually look at the tissue level at the ECG, you can see the prolongation of the T wave as well in the red here. So how do we actually measure hERG in vitro? So we actually do this at a CRO. We actually have HEC-293 cells transfected to stably express the hERG potassium channels. We use patch clamp electrophysiology recordings at the single cell level, measuring millivolt changes. We run this in eight doses in a dose response curve, and we use quinidine or terfenadine used as a positive control. And as I mentioned earlier, terfenadine, which was Celdane, is actually a good positive control in this assay. So we actually measure hERG in a couple of different ways. We actually have a manual patch clamp, which is actually a single cell assay, and this is our gold standard, where actually you have one cell in a petri dish, you take a glass pipette, you pull suction on it, and you actually have full cell recording. Once the membrane breaks open, you actually have full cell recording here. Kind of a faster version, we actually have a 384 well based plate version, which actually the cells fall down onto the micro openings, and the same thing occurs. You have suction, the membrane breaks open, and you have full cell recording that way. And again, this is how we measure these action potentials, as I mentioned earlier. So this is just an idea of what these rigs look like. The manual assay is actually run by a trained electrophysiologist. It's actually in a Faraday cage to keep out any kind of electrical interference for this, because it's a very sensitive assay. And on the right is actually the automated assay. It's kind of nice that it's all in one unit. They come in multiple well types, 96 through 84, and there's actually some 16 and 24 well assays as well. Getting into our compound X background, and sorry that it's compound X, legal made me keep it as compound X, but it is a multi-kinase inhibitor. And what we noticed is we actually had some pretty low hERG values. So our automated patch clamp came in 11.9 micromolar. When we reran it in the manual patch clamp, was what we normally do, it came back at 9.3 micromolar. And so we had some questions, and we wanted to know if the low hERG IC50 would actually translate into functional effects in vitro on human cardiomyocytes, which should encompass all major cardiac ion channels. And this is actually one of our CRO-based assays. It's the Cyprotex eCypher cardio assay, which is run in the Induced Pluripotent cardiomyocytes. And then also, the next question we had was, will this translate into functional effects in vivo in a telemetrized dog study? So summary thus far, actually, it's very quick. I have two hERG assays, automated patch clamp 11.9 micromolar and the manual patch clamp of 9.3 micromolar. A little bit of background on the eCypher-Cardio multi-electrode array assay. It's in 48 well microplates. They're induced pluripotent stem cells from our sponsor, CDI, and they're in 10% serum. Cells are a mixture of spontaneously electrical active atrial, nodal, and ventricular-like myocytes. Plating and maintenance before the assay begins takes 14 to 21 days before a stable beating phenotype is seen. With this assay, we run actually a 5-point dose-response curve in duplicate for each compound. And for this assay, we actually use quinidine as our positive control, and this one also prolongs QT as well. And compound treatment is 45 minutes with real-time tracing in all 48 wells. And data is normalized first to its baseline and then recalculated as a percent of the vehicle-treated control wells. So a little bit of background on the actual assay itself. It utilizes an Axion Biosystems MEA system, the Maestro system. And what's really interesting about this, we actually get a lot of different measurements out of here. So we get the beat period, which is the time. We get the sodium slope, which is the conduction velocity, or V over S. Our sodium amplitude, which is our action potential, our millivolt changes. We also get field potential duration, which is actually our measure of QT. And then what also this brings is we actually get to see some of these early after depolarization events, which actually translates pretty well into an arrhythmia potential. So this figure is from SI et al. And what this shows is actually really nice. Everything's on the same timeline. We actually have ECG on the top. We have a microelectrode array trace in the middle, and we actually have the action potential on the bottom. And as you can see, the prolonged QT, which is the red line, actually lines up pretty well amongst all these things, showing that there should be a pretty good correlation between the patch clamp, the MEA, and the ECG in vivo. So when we actually look at the data from the multi-electrode array, what's interesting is we have this T wave here showing here, and the little bar here is there's a QT interval which is showed here. Unfortunately, in the MEA traces, there's only a Q. It's basically a Q wave. There's no QRS complex. But we can still measure the T wave, and you notice at the right side is treatment, and the left side is baseline. You'll notice at 10 micromolar of compound X, we lose the T wave. And then you'll notice as we get into higher concentrations, the T wave is still gone. But now what we see is additional things happening. We actually see these early after depolarization events that occur, these red arrows. And also, we also see an increased beat period. You notice that the timing between the peaks here are getting longer, and that actually translates to decreased beat rate. So when we actually look at all the parameters that I mentioned earlier, so the MEC, the minimum effective concentration, is denoted by these black circles here. And you notice that for the average field potential duration, the purple line, the minimal effective concentration is 10 micromolar, which lines up exactly with our Hg values. And you notice there are also changes in other events as well. The beat period actually increases, which actually translates into decreased heart rate. We actually have a conduction velocity decrease, and we also have an action potential decrease. So these are changes that happen, although they're not happening at 10 micromolar, they're happening at 31.6 micromolar, they are still occurring. So the summary thus far is we have, of course, the Hg assays around 10 micromolar. We have the field potential duration, minimum effective concentration around 10, and then we see a lot of these other effects that occur at greater than 31.6 micromolar. So far, the eCypher-Cardio assay is matching up pretty well with the Hg assay. All right, so now the next question was, how does this translate in vivo? So we actually set up a dog radiotelemetry study, and we wanted to assess the potential acute effects of oral gavage of our compound X on arterial blood flow, pressure, heart rate, body temperature, and lead to ECG in conscious radiotelemetry instrumented male beagle dogs. We set this up, and we actually had 3 treatment groups, actually 4 treatment groups if you can count the vehicle. So we have the vehicle, we have a 5, 15, and 30 mg per kg dose groups, 4 dogs per group. And actually I put the Cmax levels here so you could actually see the micromolar concentrations that it's equivalent to. With one caveat that at 30 mg per kg, 3 animals had to be sacrificed due to GI tox. So most of the readings, all the readings at the 30 mg per kg group are just from 1 animal. So when we looked at the data, the PR interval time, there was a change at the 15 mg per kg group. There was a 20% PR interval shortening. And what was interesting at the 30 mg per kg group, that PR interval shortening at 20% was actually at 10 hours, and then by 18 hours, that PR interval shortened even to 30%. And at 15 mg per kg, the 20% shortening was at 12 hours, and there were no changes at 5 mg per kg. So when we look at the QRS interval, there were no changes at 5 and 15 mg per kg, but there was quite a interval shortening of the QRS at 30 mg per kg, indicating a change in ventricular depolarization and atrial repolarization, which is, of course, a potential for tachycardia. So when we look at the QTC, or the corrected QTC interval, using the van der Waals equation, there was no changes at 5 and 15 mg per kg, but at 30 mg per kg, there was a definite increase in QTC, indicated here, and this was at 2 hours. And of course, a lengthened QT interval is a marker for potential ventricular tachyarrhythmias, like torsade de pointe, and a risk factor for sudden death. So in summary of the results, our HERG assay results showed automated HERG IC50 of around 12 micromolar, a manual patch clamp HERG IC50 of 9.3. When we look at the eCipher cardio results, the field potential duration matches up with our HERG IC50s around 10 micromolar. We have a loss of T-wave at 10 micromolar, which actually usually precedes early after depolarization events. And of course, we see beat period decreases, sodium slope decreases, and amplitude changes at 31.6 micromolar. And of course, EAD is noted at 31.6 micromolar. When we look at the dog study, we see that there are no findings at 5 mg per kg. There's one finding at 15 mg per kg, which is the 20% shortening of the PR interval. And at 30 mg per kg is where we see all our main changes. And of course, with only one animal surviving, these doses really weren't tolerated. But there is a 30% shortening of the PR interval, and there is a shortening of the QRS interval, indicating a change in atrial repolarization and ventricular depolarization. And of course, this also is an increase in QTC, indicating a delay in the ventricular repolarization. So my quick summary slide now is that we have the HERG assays and eCipher cardio assays correlating fairly well. And then when we do the dog telemetry study, even though the doses aren't quite the same, it is predicting the eCipher cardio, the induced pluripotent stem cell cardiomyocyte assay, is predicting what we see in the dog telemetry study. So in conclusion, the eCipher cardio assay gave a FPD value of 10 micromolar, very similar to our HERG IC50 values. Other changes in beat period, sodium slope, and amplitude, as well as EADs, were identified in concentrations of greater than or equal to 31.6 micromolar. Our in vivo dog telemetry study with compound X identified changes in the PR interval at both the 15 and 30 mg per kg doses. QT prolongation was only associated with HERG blockade, was only noted at the 30 mg per kg dose. So all in all, the HERG and eCipher cardio assay were able to predict the in vivo outcome fairly well. And in closing, this eCipher cardio assay is an in vitro assay we hope to deploy when functional data is needed for all cardiac ion channels, and not just HERG, or when we also need functional confirmation of HERG effects. With that, I'd like to thank my folks at Celgene Exploratory Toxicology, Ciprotex, who runs the MEA assay, and Blake at CDI.
Great presentation, Dale. Thank you very much. Your talk really accentuated how stem cell-derived cardiomyocytes can link in vitro with in vivo results. Much appreciated. So before we move to our final presentation, we again invite you to submit questions for our Q&A session at the end of the third presentation. Simply type your question into the Q&A box on the left-hand side of your screen and then hit submit. Matt Peters is set to present the final talk of our webinar. So Matt, the floor is all yours.
So what I plan to tell you about is some work we've done in AstraZeneca using stem cell-derived cardiomyocytes to try to build frontline screens for cardiotoxicity. As most of this audience well knows, that cardiovascular tox is a major cause of drug attrition and withdrawal. And this heuristic slide is not meant to be particularly accurate, but to show you that level of concern rivals efficacy, portfolio, and cost of good failures. And this is true still, despite the fact that we have really good in vivo and ex vivo models for cardiotoxicity. And they have good translation, they're very sophisticated, And one example might be dog telemetry studies. And within AstraZeneca, we might use dog telemetry when we're down to 1 or maybe 2 compounds. And so what you get out of that is a recognition that a compound may have a hazard. And you really are left with what I would call a false choice. You either move ahead at risk or you kill a compound that you have invested many years in. And what we'd like to do to change this is to get to where we build frontline assays with the capability of driving SAR and then build safety in by design. And that might change the paradigm here. And consistent with this hypothesis, as most folks will know, is the hERG work, whereby we have now required to and routinely screen for hERG and related ion channels. And this has dramatically reduced arrhythmia. But that is a molecular perspective, and it leaves quite a bit of a gap in the functional screening area. So if we could address this, maybe we could make a shift in this pie chart on the left. So another way of showing this is to say that we have a whole host of various cardiovascular hazards which we'd like to better understand the risk, and I've shown those here at the top. And our routine frontline screens currently across the industry are hERG and related ion channels. And then we go into late phase in vivo studies. So there's quite a gap here, and if we could develop assays that would support earlier detection with diverse risk, allow us to look at mechanistic studies, develop SAR, and translate to in vivo, this could really change things. And as it happens, most folks will know that we have really good technology that is already developed and ready to go. And I've listed a number of them here, but not inclusive. And the game changer here is these assays plus the addition of the iPS-derived cardiomyocytes. They bring to this then a scalable supply that is human-based. So then you might ask, well, now that we've got these cells and we've got these assays, what is the right assay? And I get that question quite a lot, but I think it's the wrong question. It's not what's the right assay, but what's the right cascade. And that leads to very different answers when you ask that question. So when you think about a primary screen, the criteria you would have would certainly be high throughput. You want it to be frontline. You really want to avoid false negatives. So you want it to be very inclusive, and to get inclusive, you probably want it downstream in the excitation-contraction coupling cascade. You want it to be versatile. You don't want it to be limited to, say, 20 minutes or 20 hours. And you want it to have good translation. So those might be your criteria. But notice how different they are when you're evaluating something that you might use as a secondary or mechanistic study. Here, it's really critical that it be mechanistic appropriate. If you're looking at something that is affecting the mechanical contraction, you want to be measuring force, not electrical activity. So you want it to be mechanistic appropriate. You'd like this to also be distinct from your primary detection assay. You'd like it to be facile in terms of if you want to do a knockdown, you'd like that to be a possibility, and you'd like translation. So very different assays depending on what you want to do. So what I hope to cover, and this will serve as an outline, first go over what I think are among the current leading possibilities for primary screens, and then I will carry on and go through each of the various risks as outlined in the top. So let's go to primary screens. The one that is widely used currently is a calcium dye assay, and this shows data from the Flipper instrument with molecular devices, although there are other instruments out there. And this is from their publication. And you can see here there's an agent that causes change in peak width, and they can quantitate that in a concentration response mode on the lower graph. And you can see this is percent effect on the y-axis, starting at 100%. And as you get departure from that 100%, they call it a point of departure, and a 20% change would certainly be detectable. Very robust data, and it's very high throughput. So this is in 384 mode, and which would be very consistent with a frontline screen. Now, it's not the only choice. Another one that I'll highlight just because of its difference is the work from Mark Mercola's group, where they have used, again, a calcium-sensitive dye, and I think you can see that each cell, each cardiomyocyte is outlined here in a region of interest. And so they are able, through imaging, to capture each individual cell. And so you can then, from there, get ensemble views or individual cardiomyocyte views. You might want to then tease out individual cells or cells that have perhaps atrial or ventricular profiles and pull those out. This is commercially available through Valo Sciences, and you can access this as a CRO. It's very high throughput because of its either cell or population views, and it is very robust data. The second one that I think is leading the way is the impedance-based work, and I will focus mostly on that because that's mostly what I have used. And let me introduce this briefly. It uses interdigitated gold film electrodes embedded in the bottom of each well in a 96-well dish. And then the cardiomyocytes are plated to cover these in a confluent layer, and they form a resistive barrier. And that's important because a weak current is applied, and it's not enough to stimulate the cells or to be cytotoxic. It's a minimal amount of current to monitor the current path as it flows through, around, or between the cells. And as it happens, this type of technology is exquisitely sensitive, and so it leads to very robust assays. It's particularly sensitive to changes in morphology. And so one version of this technology has been developed with the Excelligence Cardio instrument. And the way that this particular version of the impedance instruments has been developed is to get data update rates of 10 milliseconds roughly. So you can see in this raw data trace, it is simply data points connected by a line. There's no fitting, and you can get very good resolution of the beating. One of the major advantages of this type of technology is that it's label-free. So there's no dye, there's no radioactivity. You can monitor the cells continuously for as long as you like. And so what I wanted to do in this next graph is show you one advantage, potential advantage of label-free. So in this, I will use a flipper dye, but first, this is in the impedance instrument. So here you can see, just for comparison, I've changed media. I've done a media change because a temperature has an effect, and you can see it's fairly stable at around 40 beats per minute. However, if I were to use the flipper-based buffer, just the buffer, you would see that there is a substantial drop in the beat rate. It drops by about half, and this has been helpful for us as we use the flipper to understand that change, that the buffer alone can have an effect because you're no longer in media. Now, if you were to add that calcium-sensing dye to this, and we know that calcium affects the contraction, then we would see an additional effect, and you can see that it drops and stops the beating by 4 hours. Now, notice there is a window there of around an hour and a half where you get good capability of detecting beating. But it is a narrow window as compared to impedance, and so that's an important difference. So overall, what assay you choose depends on how you're wanting to use it. The impedance assay has the advantage that it's farther downstream. It's detecting the actual morphological changes. It has the advantage that it's more versatile. You can read it any time you like. But it has the disadvantage that it's 96, whereas the flipper instrument is 384. Overall, then, to summarize your assay choice, I think you need to think about where you are in the excitation-contraction coupling cascade. And if you are wanting to look at changes that affect an ion channel, like nifedipine, you certainly might want to focus on something like MEA. Whereas if you're looking at something like blebistatin, which does not affect the electrical beating, but does stop the mechanical beating, then you'd want to be further downstream. And so I think I've well emphasized that the need depends on your use. So now in the last portion of the talk, what I want to do is, since I focused on primary screens, one of the criteria I set out at the beginning was it needs to be inclusive. And we've got a series of risks across the top here And what I want to do is very quickly show you some evaluations of these various risks that we've done within AstraZeneca and see if it meets some pretty basic criteria. So the first one I'll start with is an example that shows the ability to detect changes in heart rate as well as structural toxicity or cytotoxicity. And this first starts with a premise that we need to make sure that we cover, which is the relationship between force and frequency. In a mature heart, as your heart beats faster, it also beats with greater force. So a positive force-frequency relationship. As it happens in stem cells, they are immature and they don't have a fully developed sarcoplasmic reticulum, so they don't have that positive force-frequency relationship. In fact, it's inverted. And so here what I've done is used a newer version of the impedance instrument, which does have the ability to pulse the cells and thereby pace them. And so you can see at different beat rates, which I've set, as you cause them to beat faster, they beat with less force. And they do beat with less force, but here we're detecting that they beat with less amplitude. So that always assumed relationship between amplitude and force seems to be consistent. But that wasn't what I intended to show you here. What I wanted to show you is that if you understand that the negative force-frequency relationship, you can understand better some of the findings that you see. And here's one that was initially confusing to folks. Epinephrine, you know that it causes heart rate to increase and it causes force to increase. But in a stem cell assay, what we're seeing is rate go up and amplitude goes down. So that inverse relationship. Now, that would lead you to the assumption that rate and amplitude are inversely related, which they most often are. But they can be independent variables. And here is an example of a drug that causes, at low concentrations, these are the units across the x-axis are arbitrary log units. At low concentrations, the rate in blue is decreased, whereas amplitude is not changing. At higher concentrations, though, then you get a secondary effect, which is they stop beating. Then notice that cell index is a third line on here, and that's very important. It is a clear marker of a number of things, but if you have cell death, cell index will change, and it's clearly not changing here. We always include an endpoint assay as another marker for cell death, but you can see here that they are stopped. The beating has stopped without having cytotoxicity. So these two, the main point though is that rate and amplitude tend to vary inversely, as you would expect, but they can be independent. Here's an example of exactly that. This is a pacemaker current inhibitor, and as you would expect, if you block the pacemaker current, you will slow the rate of beating. Notice that the amplitude does not change initially, and this time is at 20 minutes. But if I were to reread this plate, because it's straightforward to do, at 20 hours, not 20 minutes, you can see that the amplitude very much goes up, perhaps in a compensating way. So what I hope I've shown you in one slide is that we can look at heart rate very clearly. We can see structural or cytotox internally control in real time. And I've kind of alluded to the relationship between amplitude and force, but the greater point would be next, which is can we assess contractility? And so for that, I want to emphasize that we really need to, and with AstraZeneca, we feel we need to focus on contractility because small changes in blood pressure can have a enormous lifetime risk for stroke and heart attack. So what we attempted to do was validate this assay for contractility. And this is working off some work previously by Alex Harmer, where he had validated an ion optics platform. And he had used 49 compounds, including positive and negative ionotropes. And he found with ion optics that it had fantastic validation metrics. And by that, I'm referring to some published criteria. The problem was that it's low throughput, and even more so that it uses adult dog cardiomyocyte primary cultures, which we'd really like to get away from a 3Rs perspective. So we evaluated to see if we could replace it with human stem cells. And we did it in both flipper and impedance, so I think that's worth showing. And here you can see the potency relationship in the ion optics versus the impedance data. And you can see the potencies correlate very well. Now, as for the validation metrics, the flipper data was not as good as your ion optics, but it was still in the, quote, good range. When we looked at the impedance data with this same set, it was back up into the excellent range in the 80s. So we felt like impedance was a better assay, but again, which one you choose depends on some of your criteria. As it happens within AstraZeneca, we use both. We use flipper for routine screening because of its throughput, and we also use impedance because of its performance. It is worth pausing here for a moment, though, to highlight some differences and how you can get really good quality despite some of the important differences between ion optics and impedance. Both of those have the excellent numbers on the validation metrics. One uses dog, the other uses human. One uses adult, whereas one uses fetal. One uses paced, whereas one uses spontaneous beating. And one uses imaging, whereas the other uses impedance. So we can get very good data despite these differences. And we often hear folks focus on one of these in particular, but I think it depends on your purpose. So moving on then to another risk, arrhythmia, and particularly non-HERG related arrhythmia. And for this, I'll show one slide, and it is not data from AstraZeneca, but data from Roche, Lian Gao, and Kyle Colliget while he was there, and several papers now, but I'll just highlight a couple of the key points. First, they selected a diverse panel of compounds, many more than are shown here, but you can see that the types of arrhythmias are very different within this set. What they calculated was a concentration of the drug that caused a 50% change in the beating. And when they used that metric, they found that they were able to detect compounds that were both clean and arrhythmogenic better than HERG or QT. So an integrated assay that includes all of the aspects, including the mechanical contraction, was more predictive, both positive and negative, than the traditional assays. And they clearly conclude that it has superior predictivity for detection of clinical arrhythmias. I want to point that out again. It goes back to this is good for detection, but if you wanted to follow up on it, you might want to use a different, more mechanistic study, depending on the type of cause. And I would refer you back to their papers for additional reading. Now, I want to move on and talk about a few examples of changes in blood pressure and idiosyncratic tox. And here, first I'll show the criteria that we use to evaluate this. The criteria are, can we detect it earlier than in vivo or in the clinic? Can we determine mechanism of action on or off target? And if it's off target, can we drive SAR? The first example will be a MARC kinase inhibitor. MARC's phosphorylate tau. Tau is hyperphosphorylated in Alzheimer's. So if we could stop the hyperphosphorylation of tau, perhaps we'd have the first disease-altering treatment for Alzheimer's. The problem is that Merck published some selective and potent inhibitors of MARCs, and they dropped blood pressure in dogs. And here are a couple figures from their patents, and you can see as they dose escalate, the blood pressure dramatically drops. So can we determine if this is their chemistry or if this is a target-related issue? So we synthesized a couple of compounds that were potent MARC inhibitors. We picked two that differed in potency at MARC by tenfold, and as it happens, they both stopped the beating in the impedance assay with a potency difference of about tenfold. Now, this is published, and you can look in the publication and see that by several different measures, this did not kill the cells. It simply stops the beating. But this pharmacology clearly suggests it's on target. As an independent measure, we also did knockdowns, and you can see that MARC2 and MARC4, when knocked down, also decrease the amplitude of beating. And of course, pan knockdown of marks is even more dramatic. So both of these really strongly suggest that it's on target. One further way that you can evaluate this is with a pathway agent, and in this case, something that would alter microtubule dynamics. And you can see clearly, in the paper there are a number of them, but clearly pathway-specific agents had the same effect. So we can conclude that we can detect it earlier than dog. We could drive SAR, but in this case, it's on target and we should stop. Now let me show you another example. This is check kinase inhibitors. And AstraZeneca stopped after going into the clinic and finding effects on heart beating. Schering also went into clinical trials and stopped for a similar reason. So you might suggest that this is an on-target effect. But we'd like to test that. And so we tested both of these check inhibitors and found that they both stopped the beating of cardiomyocytes without any cytotoxicity. So, to see if it's pharmacology related, we brought in a series of additional published check inhibitors, and we found that they had a wide range of potencies, some of them also stopping beating, but some of them not. So, is it pharmacology related? And as it happens, with one exception, all of these compounds have potencies in the single-digit nanomolar region. So they're roughly equally potent at check, but vastly different in their effects on beating. Now for check 2, again, no correlation. So it looks to be off target. Let's try the similar knockdown that we did for Mark. And you can see knockdown of 1 or 2 or both has no effect. So to summarize this then, what we're finding is that, yes, we can detect these findings earlier than in vivo, earlier than in the clinic. We can determine using pharmacological, genetic, and pathway-specific agents if it's on or off target. If it's off target, those assays all included error bars, even if you didn't see them, and so it was very robust, and we could drive SAR. So I want to tackle in the last bit one final risk, and that is around kinase inhibitor cardiovascular tox. And many of you may know that among the FDA-approved kinase inhibitors, many of them are tyrosine kinase inhibitors, and many of them are cardiotoxic. So what we wanted to do is see if we could begin to sort of deconvolute the kinases that contributed to cardiotox. And I will only take the two most extreme positions here, and the rest of it will be published shortly. But those two extremes are that it is not a particular kinase, it's all about promiscuity, or the other extreme would be that it is only particular kinases. So first, in the promiscuity hypothesis, what we have done here is we have worked on the shoulders of Ambit, who very generously published 65 compounds with structures and identity, and with selectivity profiles across almost 400 kinases out of almost 500 in the kinome. And so then these compounds come with, by definition then, a selectivity score. And one hypothesis would be that as you go have more promiscuous compounds moving to the right, your beat rate would be more affected. There is just a promiscuity correlation. Or it may be that there is a threshold. Once you hit a certain number, you have an effect on beating. Or there's no effect, that it's all about individual kinases. Of course, there would be a slight trend, but not a clear correlation. And so Sarah Lamore, who did this work as a postdoc here, what she found was that there were some compounds that were quite selective, yet had effects on beating, and other compounds that were quite promiscuous, but did not have effects. So the promiscuity score was not a sufficiently useful tool. She did go on, and it'll be in the publication, to look at each family. Tyrosine kinases were not correlated. In fact, they were the least correlated. But what she went on to do is take the other extreme, which is, are there particular kinases that are correlated with effects? And the answer was she found 46 that were, and here I'm showing that she knocked down each of those 46. And of those that she got at least 75% knockdown, they're shown here, And 13 of those kinases, when knocked down individually, affected beat rate. The identity of these will be revealed at the publication. And 19 different kinases, I might add, when knocked down individually, affected amplitude of the beating. So why did we not get all of them? Well, it might be that some of them were polypharmacology hitchhikers. An example might be something like Mark I and Mark II. Maybe we don't have the compounds to distinguish between Mark I and Mark II, so you can't tell which is the real contributor. Another possibility for not all of them validating is that perhaps 75% knockdown is not sufficient. Another possibility is that they are convergent. Trying the Mark example again, maybe it's that Mark I and II are both correlated, and it requires that they are both knocked down. So the way I would interpret this data is that the individually confirmed by knockdown are clear positives, and they contribute. Those that didn't confirm by knockdown may still be of value. So wrap up by saying that we started with some really diverse risks, and we started with a real gap for primary and secondary screens. And where I think the field is moving to is we are able to detect. We have the capability to detect these diverse risks, and we can move them earlier. We can follow up on them both in mechanistic as well as SAR studies. But your choice is driven by your need, whether it be primary or secondary screens. So with that, I'd be happy to take questions as all of the presenters would.
And thank you, Matt, for a wonderful presentation. You gave the audience an exquisite look into cardiotoxicity screening methods that will no doubt become an invaluable part of the drug discovery process in the very near future. Thank you again. Okay, we've received a number of very interesting questions from our audience, so let's take the first one.
Okay, so let's get to our first question. The first question is for Blake. Blake, one of our audience members said they heard there's a newer version of iPSC-cardiomyocytes available. How are they different from what was presented here?
All right, well, these cardiomyocytes, the newer version are called the iPSC-cardiomyocytes squared. And this is another version of the cardiomyocytes, made from the same starting material, but it's ready to use after you thaw it much quicker. So these are typically ready, these new cardiomyocytes are typically ready within 4 days of thaw rather than a week or two post-thaw. So it's really the same cardiomyocyte material. We've just optimized procedures for handling and quicker use.
Okay, great. Thanks. So the next question is for Dale. Dale, this is sort of a two-part question. One of our audience members wanted to know, is it total or free Cmax that you were speaking of, and what is the effect on heart rate?
So thanks for the question. The Cmax noted is the total Cmax. So the compound is highly protein bound, so this may be a reason why the in vivo concentrations at which we see the effects are much higher than our in vitro concentrations. And interestingly, to answer the second part of the question, the heart rate in the dog telemetry study actually increases at 12 hours as opposed to decreases like in the MEA study. And that's at both 15 and 30 mg per kg. And additionally, in addition to the increasing heart rate, there's also a concomitant decrease in pulse pressure at both of those higher concentrations.
Okay, great. Thanks, Dale. So next question is going to be for Matt. Right here. Oh, here it is. Matt, you mentioned that tyrosine kinases appear not to cause cardiotoxicity. Can you explain that a bit?
Sure. It's not that they don't cause it. It's just that as a group, we thought that the kinases that correlated with cardiotoxicity would be most concentrated within the tyrosine kinases. That was a hypothesis, and that's not what we found. In fact, as a group, they were the least correlated, rather surprisingly. But that doesn't mean that there aren't individual tyrosine kinases that are perhaps very causative. So it's a bit of context there.
Okay, great. Thanks, Matt. Blake, it looks like we have another question for you. One of our audience members said, you mentioned mechanical properties of the eye cells being phenotypically appropriate, but classical cardiac people are always asking about presence and function of T-tubules for correct contraction phenotype. Do your eye cells have differentiated T-tubules?
Yeah, that's a great question. Thanks. Typically, the short answer is that the stem cell-derived cardiomyocytes, when cultured on that 2D non-yielding tissue culture plastic, they have the T-tubules, but they are not as fully developed as one would find from a freshly isolated prep. So they are there for phenotypically correct interrogation, but they're not going to be as fully developed as one would find in the freshly isolated preps. What we are seeing, though, is when maybe I would call them these second-generation culturing techniques of whether it's patterning or using 3D type of pattern structures, we're starting to see more and more morphological correlation with what one would expect. So really we're seeing limitations in the culturing techniques that are being overcome by these second-generation culture developments. So thank you.
All right, great. Thanks, Blake. So our next question for Dale again. Dale, what is your normal cardiovascular toxicity screening paradigm?
So what we normally do is we actually use the automated patch clamp as our screening tool, which allows us to bin the compounds based on their hERG inhibition. And we actually have a cutoff of less than 10 micromolar we consider high risk. And then from 10 to 20 micromolar, it's kind of it's possible risk. But that doesn't necessarily mean it kills the compound, so it can go through the system, and if it becomes a higher value compound, then we'll follow up with a manual patch clamp and also run the MEA assay. And then as we get closer to maybe a short list candidate, we'll even go even further into risk using a dog telemetry study if it's really high value, and we're just needing to make sure that what we're seeing in the MEA and the patch clamp translate into in vivo.
Okay, great. Thanks, Dale. So Matt, we have one more question for you. One of our audience members wants to know if you will publish the identity of the kinases associated with functional cardiotoxicity.
Yes, indeed. That's being worked up now and should come out shortly. It will include both those kinases that appear to be causative as well as a subset that will make up a predictive model.
Okay, great. Thanks, Matt. And looks like our last question is going to be for Dale. Dale, how does the MEA assay translate to human cardiovascular injury?
Good question. So what we use the MEA assay to predict only our in vivo tox models, such as our dog telemetry studies. But we feel that since the dog is a very good cardiovascular tox model, the translation to humans should be there. However, we haven't run compounds in their system yet that have been shown to have human cardiovascular tox issues. But one of our goals is actually to run some of those actually through the MEA and see if it actually correlates with what happens in people.
All right. Thanks very much, Dale. And with that, we've come to the end of our webinar. I'd like to remind everyone that the webinar will be archived for 6 months on our website at www.genengnews.com. So if you missed parts of it, you can watch it again, or feel free to forward the link to your friends and colleagues. I'd like to thank Blake, Dale, and Matt again for their informative presentations, and I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks to CDI Technologies for sponsoring this webinar. So hopefully we'll see you again in another Gen webinar in the near future. So goodbye for now.